Hey there, science students, and happy Monday. Today is Monday, February 8th, 2021, and I hope you are doing well. Today, the focus of our class is going to be on forensics and forensic science, but I want to take a moment and just remind you to complete the chart on page 12 of your LearnEd notebook. It is entitled Biotechnology Tools. And when you do the recorded session on YouTube for the entire booklet, you will also hear the same information. So if you've already done the chart, you can go ahead and skip over this part. But if not, I just want to reiterate, you've got 3 columns on page 12. You have the first column is about cells. The second middle column is about genes. And the third column is proteins. And for each of these columns, each of these terms, cells, genes, and proteins, in the first box, I want you to describe what they are. So for the first one, cells, you can get all the information from this page here. How are they used? And then how do they benefit society? So you're going to get all the information that you need to fill out the first column on page 12 using this slide. The next slide, which is genes, right here, you're going to go ahead and, again, fill out the middle column. What are genes? How are genes used, and how do they benefit society? And then the third and final column is proteins, is for proteins. So proteins, what are they? Here we go. You've got some information right here. How are they used? Right, here you go, right here. And then finally, how can they benefit society? So use the information for each of these 3 slides to go ahead and complete those 3 columns on page 12. And now we're going to move into forensics and forensic science. This is an exciting field. It is changing and growing on a daily basis. Many of you have told me that you're interested in going into this field of study after high school. So it is very exciting. So forensics is basically using science to investigate crimes or crime scenes. Biotechnology in crime scene investigations includes DNA fingerprinting. Now, this is not the type of fingerprinting that you use from your fingers when you put your finger on the ink pad and then, show your fingerprint. This is actually DNA fingerprinting using your DNA material, right? It basically shows that you are you, right? Remember, only you has your DNA, your set of DNA, unless you have an identical twin. And in that case, you guys would share the same DNA. So DNA fingerprinting includes analyzing and comparing DNA fragments. So it is created in the lab. Scientists use this DNA to create, they separate the pieces through a process called electrophoresis. And this is the separation of DNA through an electrically charged field. So you're probably familiar when you think of DNA and examining DNA, you see these bands. These have been separated using electrical charges in a process called gel electrophoresis. So restriction enzymes are enzymes that break things down. These are used in the process to cut DNA into smaller pieces. The smaller fragments travel farther in the gel. Each person's DNA has the same banding pattern. We're going to take a little look at that here in a second and let you put that knowledge into practice. So the following is a DNA fingerprint produced by gel electrophoresis. Who do you think is the culprit, or should I say, whose DNA was found at the crime scene, according to the gel below? So if this is the DNA that was found at the crime scene, let's look at that for a minute. We have a single band, a single band, a double band, and a double band. Now let's compare this pattern to suspect 1's pattern, suspect 2's pattern, suspect 3's pattern, suspect 4's pattern, and finally suspect 5's pattern. Take a minute and look at that banding pattern and see which suspect matches the DNA that was found at the crime scene. If you guessed suspect 3, you would be correct. We see double, double, single, single, just as what was found at the crime scene. So we could say that suspect 3's DNA was found at the crime scene. Now, does that prove guilt or innocence? That is up to the detectives and all the people doing the investigation. That's up to them. But we can prove, looking at the evidence, that their DNA was found at the crime scene. So DNA is also used for other purposes besides deciding or proving if someone was at a crime scene or not. It can be used for paternity testing. Paternity, as opposed to maternity, which is the mother, paternity is looking for who was the father of an offspring. So it identifies similarities in the DNA of parents and offspring. It can also be used to catalog endangered species. So think about zoos and aquariums. Forensic science, DNA patterns are very beneficial to organize the information and discover unknown information about a species and to even identify species or their parents. For example, you might want to know the bloodline or the family tree of a band of gorillas. If you're studying gorillas in the wild. So this is, as you can imagine, studying and analyzing the DNA of humans and other organisms is hugely beneficial. So now what I have for you guys, now that hopefully I've enticed you, this is an exciting field, and it's science and it's mystery and it's conservation all rolled into one. Maybe you're interested, if you weren't before, in becoming a forensic scientist. What I'm going to do is I'm going to take you on a little field trip as best as we can do virtually. I'm going to introduce you to a young lady named Sarah Finch, and she is a forensic science technician with the city of Fayetteville. So she lives just down the road, about an hour away in Fayetteville, North Carolina. And this is what she does. Take a minute and watch with me, and let's see how exciting Sarah's job is, her career choice. So let me go ahead and share with you. This short video. It's only about 4 minutes long. So let's go ahead and take a peek and see what Sarah's career is like. Here we go.
I found out that I had an interest in forensics since I got my elementary school. Learned about it in popular media, different TV shows, and first reading forensic files on that. But after looking more into the field and figuring out exactly what they were, I decided to enter the state program, which the forensic biologist. So now I'm still having classes to the field work. But I don't want to do this for people since I was a kid since then. The most interesting thing about it is probably what I went to actually go and stay with him and study undergrad. And then I went to the big part of the investigative process of medical journals. I worked on anything from mammograms, so anyone that has evidence all the way to homicides, it really depends on the circumstances of the case. So we're very fortunate as well what we do. Any crime scene we're going to talk about what we see in the form of notes, sometimes in the form of photography, genetic knowledge, some fingerprints, possibly DNA, just such a measure of processing, we can do that. And all the other parts of the things that we see. So we can look at personally, but the thing is with the analysis is kind of similar, but from the best understanding of what was in front of us, again, as so we're talking about some questions that DNA and fingerprints. From a central evidence that the suspect came out during the incident. So one of the
And that's what it's all about, guys, isn't it? Right? Helping each other. So I hope that you enjoyed spending a day in the life with Sarah Finch, a forensic technician from just down the road in Fayetteville, North Carolina. I would like for you to make sure that today that you have your notes completed, the chart on page 12 that I have explained how to fill out, and also the forensic notes on page 13. Okay, and we'll talk more about this later in this week when we talk about careers in biotechnology. Thank you so much, and I hope you have a wonderful Monday.